So welcome back for more AP Biology. We're going to continue our little chat of enzyme-ish, chemistry-ish things with talking about how do we living things, gather massive amounts of energy. And the first of those is what we call photosynthesis. So we're going to look at the role of enzymes in photosynthesis, and then how do we store away that extra energy. They test on this. It's confusing. So when we look at metabolism is one of those catch-all words that just turns out to mean the chemistry of living things. And what you can do with metabolism is we can take any of these molecules that you see on the screen. So ribulose 5-phosphate or cytidine or uracil or tryptophan or guanine or glyoxylate or cysteine or alanine or whatever thing you want to deal with that's on here, acetate, glycine. And what we have is a series of chemistry tricks that we can do that connect all of these pieces. And all that chemistry we collectively call metabolism. If you look in the bottom corner, there's a whole bunch of things like that says like Krebs cycle and amino acids and lysine loops and stuff like that, and glycolysis and the pentose phosphate shunt. But we have a whole bunch of ways that we can manipulate molecules and convert between different things. That's just what metabolism turns out to be. Metabolism has to follow this series of laws that we call thermodynamics. So there are 4 laws of thermodynamics. They're just mathematical statements. You don't get to argue with them. They just are what they are. For the most part, we don't care about the 0th and the 3rd law, but we care about the 1st and the 2nd. The 1st law says within a closed system, meaning nothing gets in, nothing gets out, you just have a total and that's all you get to play with. Earth turns out not to be a closed system, therefore first law of thermodynamics does not apply to us. The 2nd law of thermodynamics is what we call the entropy principle or the efficiency principle or the chaos principle, which is nothing is efficient, no chemical process, any process, physical, chemical, whatever, biological, is efficient, and thus you're going to convert your energy from a useful form to a useless form. And that useless form usually will increase entropy or increase chaos or increase randomness. That's what we have to fight as living things. So the easiest and most abundant energy source for us would just be go up and look at the sun. The sun is beaming a whole bunch of light or electromagnetic radiation at us, and what it does is it's sending us light in the form of two ways. One of those ways that it's going to send information to us is in waves. Therefore, we have a wavelength and we have frequency. The other way is as distinct little bundles or bullets that we could call photons. So it's particles and it's waves. So it behaves kind of strangely. What we notice when we analyze the type of light that exists is we humans can only see a very narrow band of all of that exists. So when we sit there and say, oh, look at all that we can see, we don't get to see that much. We get to see basically between 400 nanometers and 700 nanometers. But there's a lot more to light than just that. It's just all we get to see. We also notice that the amount of energy in light isn't constant, meaning light that has very large wavelengths or very low frequencies don't have as much energy as light with short wavelengths or high frequencies. And that turns out to be important in just a moment. Meaning red light has less energy in it than purple light does, or violet light. So we have all this light that's coming at us as living organisms, and one of our challenges is, let's capture it. Can we use it? Well, for those of you who have ever gone outside when it's summertime and you're wearing all black, what do you notice? It gets really hot really fast. Well, why is that? Black absorbs light, and the result is you're absorbing all the light that's coming at you, and it gets really hot really fast. If I look at a plant, any type of plant, so it could be a tree or a shrub or a moss or hornwort or a liverwort or an algae, what have you, some type of photosynthetic organism, something that can take light and use that to form energy, what we notice is they have lots of different colors in them, lots of pigments, lots of things that can absorb light. And what you can form is this thing here that we call an action spectrum, which shows us How much do each of these colors, so chlorophyll A, B, carotenoids, phycoerythrins, phycocyanins, how well do these things all absorb different wavelengths of light? And what you get is something that looks like this. Collectively speaking, if we add all the pigments up, what do we see? We absorb all the light that we can visualize because they want to absorb as much light as possible. All of this stuff turns out to be part of the thing that does the photosynthesis, the making of energy from light, and that's called the chloroplast. When we look at the chloroplast, it has two membranes, an outer membrane and an inner membrane. The outer membrane turns out to look shockingly like a eukaryotic membrane, and the inner membrane looks shockingly like a prokaryotic membrane. We get these stacks of what look like coins on the inside called thylakoids. An entire stack is called a granum. The space inside of each thylakoid is called the thylakoid space. Genius. It happens to have its own DNA. It turns out to have its own set of ribosomes, whatever those things are. The space between the inner chloroplast membrane, not mitochondrial membrane, inner chloroplast membrane and the grana is called the stroma. But if I really looked at the thylakoid membranes turn out to be the exact same thing as the inner chloroplast membrane. And that space within the thylakoids, that thylakoid space, turns out to be the exact same space as that gap between the inner and the outer chloroplast membranes. That might mean something. I have all these folds all over the place, which means I have a lot of surface area, and having a lot of surface area probably tells you something about its function. So if I look at photosynthesis, photosynthesis has two parts. We have a part that's required and a part that's not. The part that's required is referred to as the light-dependent reactions. They absolutely require light, and it's the only part of photosynthesis that is absolutely necessary. What do you have? We have electrons being moved around different proteins found in that membrane, that thylakoid membrane. And as those electrons are being moved, they pump protons. So there's a proton pump, and it turns out to move stuff from the stroma, the inside, out of the stroma and into the thylakoid membrane, or the lumen, as you can see here. When you do that, we're building up a concentration of these protons. So we get a lot of protons inside of this lumen, not that many on the stroma. We're having a buildup of positive charges in the lumen, not that many in the stroma. Concentration difference and a charge difference, that's called a membrane potential. We've dealt with that before. We're building up a membrane potential. Well, what's building up this membrane potential? There turn out to be two things inside of this system called photosystem 2 and photosystem 1. It's not important that you know their names, nor is it important to know that photosystem 2 shows up before photosystem 1. These are complexes that have proteins and they have pigments that can absorb light and pass it all into one spot. They can suck up light and shove all that energy into one spot, into one electron. And that electron then reacts with a whole bunch of other parts that make it move from photosystem 2 ultimately to photosystem 1 and then to off wherever it's going to go. So we get this flow of electrons. And what's driving this flow? Light is. Light is powering the motion. And that powered motion of those electrons makes a proton gradient. But what are we going to do with this proton gradient? We have a membrane potential, but we're not doing anything. There's a structure very much like a wind turbine or a water wheel that if you were to take a wind turbine, blow wind through it, it twists and you produce energy. If you have a water wheel, water dumps through it, it twists, you produce energy. So there's something about twisting motions that produce energy. It turns out there's an enzyme, a structure that makes ATP. So it's the ATP making enzyme or the ATP synthesizing enzyme. What would we call that? Let's call it ATP synthase. Maybe, perhaps. What this thing has is a channel in it that allows protons to flow through it. And as the protons flow through, the turbine starts to twist. And it takes 3 clicks to make one full turn, and that turns out to be just enough to make ATP. So I let a stream, a waterfall of protons move through this thing, it twists around, and we produce ATP. Genius. We just have 2 questions left. Where did we get that electron from, and where did it go? Let's do the first one first. Where did we get the electron from? Well, we need a source of electrons. Plants, or at least the first ones of these things that undergo this process of photosynthesis, most likely evolved in water, which means they were surrounded by water, so water should be everywhere. So what if we took water and we turned that into electrons? So, turns out on one of the photosystems, there is a complex there that can rip water apart to produce protons, electrons, and this waste that we'll call oxygen gas. And the oxygen gas will just leave and it'll go into the atmosphere. And if you let this happen for 1 billion years, we go from a 0% atmosphere of oxygen to a 20% or 25% oxygen atmosphere. Kind of genius. Plants did that to us. That's the first question. Where did the electron come from? Second question, where did it go? At the very end of the system, we blast it with more energy in that second photosystem, and then the question becomes, well, where are we going to put it? Well, there are things that exist in chemistry or metabolism chemistry that we call electron carriers. One of the famous ones is called NADP. And NADP is just, it has a name this long. And all it does is it sucks up electrons. So if you add an electron to NADP, the electron gives it a charge. We don't like that, so let's add an H to it. And we get NADPH. And what that is carrying is a high energy electron that we can then dump somewhere. And we can do something with it. Voila, we have photosynthesis, the end. It turns out that this doesn't need to go in that straightforward manner from photosystem to photosystem. There's a version of this that bypasses and we just kind of go in a circle. Question is, well, why would we do that? Excellent question. There turns out to be an analog. There turns out to be an analog to this with cell respiration that will tie back to hormones that we'll talk about later. There's an enzyme that's also found inside of these plants called rubisco. Rubisco stands for ribulose 1,5-bisphosphate carboxylase oxygenase. And this enzyme turns out to be one of about 3 on Earth that are capable of fixing carbon. Fixing carbon is a fancy way of saying I can take carbon, which is useless, slam it into something, and now make it useful. This is one of those few enzymes. It also turns out to be really horrible at its job, but, devil's in the details. So, what if you have too much ATP? You produce a lot of ATP through the light-dependent reactions. We have lots of NADPH. And now we want to store it away for a rainy day. How do we do that? There's a process called the Calvin cycle. The Calvin cycle occurs in the stroma of the cell, which is outside the thylakoid membranes. And what it does is it takes a molecule, a 5-carbon molecule that we call RUBP, ribulose 1,5-bisphosphate, and through the help of that enzyme, rubisco, we can grab a carbon dioxide and attach them together. That gives us a 6. Turns out this 6 is unstable, so it will fall apart into a couple of 3s. This one here, they show you 3-phosphoglycerate. I can then react it a couple of times to end up making something I call glyceraldehyde 3-phosphate. And it turns out this thing, G3P, glyceraldehyde 3-phosphate, is capable of being turned into glucose. I could also turn glyceraldehyde 3-phosphate, a 3-carbon molecule, using a whole bunch of funky chemistry. I could turn it back into RUBP, the ribulose 1,5-bisphosphate, and this cycle will continue. Well, how does this use the leftover ATP and the NADPH? Well, as you look at the picture, in order to drive this process, I have to utilize ATP, turning ATP into ADP. I also have to use NADPH and turn it into NADP. Because if I do that, the result is I can now keep going and I can store all that energy inside of sugar. I didn't really tell you how we turn a 3-carbon back into a 5, and this is why. Moving on. Carbon fixation turns out to be a problem for plants because rubisco is pathetic at its job, like I said. It evolved in an atmosphere that had carbon dioxide, but it never had oxygen gas. So it doesn't know how to play with oxygen gas. So the result is plants have 2 different ways of solving this puzzle. So if you're in an environment where you don't need to worry about confusing carbon dioxide and oxygen, you just go forth and you do the Calvin cycle. We call those plants C3 plants because the first thing that you make is a 3-carbon structure. If you live in an area where we have to worry about confusing oxygen and CO2, what we do is we separate out the location of the Calvin cycle and where we grab onto the carbon. So we have a spatial difference between Calvin cycle and getting a hold of our carbon dioxide. That process is called a C4 pathway, and that occurs in like bamboo and sugarcane and stuff like that. You'll find that in tropical areas. But we have another problem from your transpiration lab is, what about water loss? So what if we live in an area that's really hot or really windy? How do you prevent water from escaping these plants? Because where do you get the CO2 from? You have to open up the plant, open up the holes, open up stomata, and CO2 goes in. But water can then leave. And how are we going to solve this? That is solved in types of plants called CAM plants, or crassulacean acid metabolism. CAM, you call them cacti. What they do is they separate carbon fixation and Calvin cycle not by distance or by location, but by time. They'll grab a hold of carbon at night, and they'll do the Calvin cycle during the day. And both of these, C3 or C4 and CAM, are ways around issues that show up with carbon fixation.